All right, good morning, grade 10s. Well, at least it's morning for me. When I'm recording this, who knows what time of day it is for you as you watch it, but thanks for watching. So we're going to do chapter 8, at least a good chunk of chapter 8 in this video, and we're focusing on the cell membrane. So let's get started. You already know that the cell membrane is responsible for what's coming in, going out of the cell. So it separates the living cell from its non-living surroundings. You could kind of think of it as something we could refer to as a biobarrier. It's very thin. It's only about 8 nanometers thick. It's controlling what goes in and out of the cell. It's what we call selectively or semi-permeable, which means some things are going to get through more easily than others. And lastly, we have to talk about these terms hydrophobic and hydrophilic, because one part of the membrane likes water and one part of the membrane not so much. And it's made of a molecule called a phospholipid, and we also have proteins, and there's other macromolecules as well. So let's look at this thing called a phospholipid, which is definitely the primary molecule that makes up the cell membrane. It has a phosphate head. If you can remember, that phosphate is a PO4 3- polyatomic ion, and it has fatty acid tails, 2 of them. Now it's the fatty acid tails that are hydrophobic, that don't like water so much, whereas the head is hydrophilic, perfectly fine with water. The membrane itself is arranged as what we call a bilayer of these molecules. So there's 2 layers of phospholipid molecules, and they're going to arrange themselves with the phosphate heads facing out towards either the outside of the cell or the inside of the cell, because it's primarily water. Remember, they're hydrophobic, they like water. Whereas the fatty acid tails, they don't like water, so they end up pointing inwards. And there you go. It's sometimes analogous, and remember, it's 3-dimensional. This will go all the way around the cell. So it's sometimes the analogy. It's like where a bunch of bison or buffalo bison would form a circle, and their heads would face out for protection, right? So it's a little like that. That's used as an analogy. Now. Here we go. Now, let's talk about these terms polar and nonpolar. The hydrophilic heads are polar. Now, remember, that has to do with charge distribution within a molecule. An unequal distribution of charge leads to a polar molecule, and so the phosphate heads, remember, are negatively charged, whereas the fatty acid tails are nonpolar. And that also leads to them being not liking water so much, because remember, water is polar, and like dissolves like likes like. So again, those hydrophobic tails, nonpolar, still not liking water. In 1972, we have Singer and Nicholson. They proposed that there are also membrane proteins embedded in that phospholipid bilayer. That leads to what we call the fluid mosaic model. So again, you can kind of think of these lipid molecules like in motion. They're like kids in a mosh pit, kind of at a concert. They're jostling with the other phospholipid molecules beside them. But there's also these proteins embedded in the phospholipid bilayer, but they're not stationary. They have the ability to flow and move, and they're random. Hence the mosaic part of the model. Because a mosaic, if you had a floor tile mosaic, it wouldn't be like a checkerboard, like a really set pattern. You'd have some black ones here and there. And then imagine that everything is kind of fluid and bendy and in motion. You have the fluid mosaic model. No, that's fine. All right. So this is a slide that just kind of has everything as an artist would render it in terms of some proteins that go right the way through, some proteins that are just more surfacey. This would be the inside of the cell. This would be the outside of the cell. We also have some other macromolecules. These are representing carbohydrate strands and cholesterol as well. Okay, so let's talk more about these proteins here. Remember that cell membrane and organelle membranes each have a unique collection of proteins. The membrane proteins may be peripheral, which means simply to be kind of on the outside. They might be loosely bound to the surface of the membrane. They might act as identity markers, things we call antigens. By the way, that's what would give you your blood type of your red blood cells. They have particular markers or proteins on their surface, which we call antigens, and they give you the blood type that you happen to have. We also have integral proteins, and integral proteins might penetrate the lipid bilayer. They might go across the whole membrane. They're quite often used in transport, and we're going to be talking about that as we go through this chapter. Some of these proteins act as what we would call channels. So they kind of open up a way for molecules to move either in or out of the cell, or some of them act as pumps. There's many functions for membrane proteins. Transporting, which we're going to talk a lot about, but some of those proteins are going to act as enzymes. So they cause a change, chemical change within the cell. Some act as receptors, right? So like hormones that flow through the blood, they need to act on only certain cells. So only certain cells are going to have proteins that act as receptors. Just like your cell phone is just designed to pick up a particular wavelength and react to it, because that's what it's programmed to react to. So it's kind of the same idea. Again, we talked about the identity marker, okay, so that your body knows which cells belong to you. You have some proteins that are they're like an ID pass. Some of those proteins are going to actually help cells to stick together. And then remember, we have this cytoskeleton, sort of the scaffolding network of protein tubes that help maintain a cell's shape. Well, some of the proteins act as anchor points for the cytoskeleton. All right, so that was section 8.1. Now in 8.2, we're going to start to talk about transport. So one of the things we need to review is a concept called diffusion. So some substances can simply diffuse, which is going to mean that they're going to travel from a high concentration to a low concentration. Now remember, the cell membrane cannot be impenetrable. We have to allow some things in and not other things. So what are some things that we want to come into the cell? Nutrition, food, in the form of carbohydrates, proteins, sugars, amino acids, lipids, some salts, oxygen, water. And what are some of the things that need to be removed from a cell? Well, we have waste products like ammonia, salts, carbon dioxide, water. We're going to get to why water moves in and moves out. And anything that the cell happens to be producing, because cells are like little factories. They're often making something, and whatever they make often needs to be able to leave the cell. All right? Now, how does this happen? Well, let's come back to diffusion. So diffusion works on the fact that molecules are in constant random motion. We often call that Brownian motion. Over time, they will distribute themselves evenly. That's just a universal law, often called entropy. Small hydrophobic molecules, remember hydrophobic, they don't like water. These molecules can diffuse in and out of cells quite readily. And so they're going to just move from a high concentration to a low concentration. Okay, so you see here the molecules are tightly together, but or maybe they're inside the cell tightly together, but because of that random motion, they're going to spread themselves out evenly. so that you'd have an even concentration inside the cell as well as outside the cell. Again, it's based on the second law of thermodynamics, which we know can be said in many different ways. Remember, its basis is that, remember the first law says energy can't be created or destroyed. It can simply change form. But the second law says that when it changes form, some energy is lost, and that's again just due to often randomness. Or another way of stating the second law is it's called the law of entropy. Particles or things in this universe tend to go to the lowest state of energy, and part of that is just because of their random motion. The universe tends towards disorder, right? I often use the example of a deck of cards. When you get a brand new deck of cards, that's very orderly, but we know that as you play with that deck of cards, it gets all mixed up. And you would never expect in your wildest dreams that you would go after taking a deck that's been used and used and used to use that deck once again, and then turn it over and look and have it perfectly the way you bought it, perfectly sorted in suits and numerically. We know that would kind of never happen, right? See, so that's that idea of randomness. It's why your headphone cables and cords get all tangled up too when you put them in your pocket. All right, so for example, if we had some molecules of dye over here and then we had a membrane and the dye is in water, we know as long as the dye molecules can fit through these spaces, that if you give them time, they're going to disperse. They're going to spread out just because of the random Brownian motion until a state we call equilibrium. Now that doesn't mean they stop moving. It simply means that maybe for every dye molecule that moves to the left, there's a dye molecule that moves to the right. Okay. So again, diffusion, its basic definition is movement from a high to a low concentration. Okay, there they go. And diffusion, guys, can happen. There doesn't need to be a membrane for diffusion to happen. You know, if somebody brings a bowl of popcorn or is making popcorn in one area of your house, it doesn't take too long for the smell of that popcorn to diffuse throughout the whole house. There doesn't even need to be a barrier for those molecules to go through. It's also considered a form of passive transport. Passive meaning it doesn't require any energy to make it happen. Okay? No energy needed. Okay. Now, we're going to end this lesson, or end this video, by talking about the movement of water. So the movement of water can also move from a high concentration to a low concentration through the cell membrane. It's now, part of the problem is that it's polar, and remember we said non-polar molecules or hydrophilic molecules could move quite easily. So actually, even though water can simply diffuse, and by the way, we would call that osmosis, some water is capable of moving straight through the cell membrane. Some of those proteins called aquaporins, they make channels so that much more water can flow through the membrane. But it's still osmosis. Okay, so of course water is very important to life. And like we just said, water can diffuse from a high concentration to a low concentration across a semi-permeable membrane. So osmosis does require a membrane. Whoops. I just wanted to look at this U-tube, for example. So in this U-tube, we have water, but then we also have some solutes. And we can tell that at the start of the experiment here, we have a lot more dissolved solute, so maybe like salt. A lot more dissolved salt on this side than the other side of this barrier. Now we're going to assume that the particles of solute cannot cross the barrier. This time what can cross the barrier is the water. The water is represented by the blue liquid, right? So water, here we have lots of water, you can see it, compared to this side. So we expect that the water will move from where it's highly concentrated to where there's less water. And so actually over time, we'd see the water level rise on the right-hand side of this U-tube. All right? That's a water pressure thing. So direction of osmosis is determined by comparing the total solute concentration from each side of the barrier. Now we do have some terminology for different solutions when we're making these comparisons. One of the terms is hypertonic. So hyper means more, right? More what? More water? No, not more water, more solute. So this could be represented by a very salty solution. We could consider that hypertonic, but it's always a relative term. It's always comparable to something else. It's like using terms like warm or cold. Those are relative terms, and it always depends on what we're comparing it to, right? Because for us in Calgary here, we can say we're having a warm winter day when it's 10 degrees Celsius outside. But if you were in like LA, for example, and it was 10 degrees, you'd be freezing. You'd want to walk around in your parka, right? So that's what I mean by relative. Now, so hypertonic means lots of dissolved solutes, like a very highly salty solution, which means there's less water, right? Hypotonic, hypo means less than, so there's less dissolved solute compared to another solution that you're comparing it to, which would mean relatively more water. Then isotonic would mean equal solute concentrations. Same amount of dissolved solute, all right? So if, for example, we look down here in this blue rectangle, on the left we have a hypotonic solution. So the yellow represents dissolved solutes. On this side, we have the hypertonic solution. Lots more yellow, lots more dissolved solutes. Which way is the water going to move? So the water is the blue component of the rectangle. Well, it's going to move from high to low water concentration, or from the hypotonic side to the hypertonic side. So water should be flowing to the right, because it's actually trying to balance out the concentrations. And there's just lots more water over here than over here. So water likes to flow to the hypertonic side. So there we go, water's moving to the right. Again, guys, cell survival depends on this whole balance. Here we have a slide that's comparing animal cells with plant cells and seeing which solution that they're actually designed better for. So if we started off with putting these cells in a hypotonic solution, so that the solution compared to the cell has a lot less dissolved solutes, which remember means more water. Water wants to move into the cell, right? Now for animal cells, not so good, right? Because all we have is a cell membrane, and if lots of water is flowing in, if too much water flows into an animal cell, it could actually explode. However, plant cells love water, and they have that cell wall for added structure and support. So actually, plant cells really like this being in a hypotonic solution. It gives them what's called their turgor pressure, and it keeps them nice, full, and firm. Now, if you put an animal cell in an isotonic solution, that's kind of just right. The animal cell keeps its shape, water comes in, and when water comes in, it might be bringing materials that the cell needs with the water, and then water can flow out. And when water flows out, it might take away the waste products. And everything stays pretty normal. Plant cells can deal with isotonic solution, but it's just not ideal. So we still have survival for the cell, right? Because we have this balance of water in and water out, but it's not going to have much turgor pressure. So this is when plants get wilty, when they don't get enough water, and the soil becomes more isotonic. Then neither cell really likes a hypertonic solution. This would be like putting both cells into a very salty solution. So the animal cell would lose a lot of its water, right? Because again, water is going to move to the hypertonic solution, which is outside the cell. So the cell can shrivel up. And the plant cell, it kind of maintains its shape because of that rigid cell wall, but it becomes what we call plasmolyzed because it loses way too much water. Its central vacuole shrinks, and that's just not going to be a successful plant if those conditions stay that way. Okay, so there we go. Fresh water, a balanced amount of solutes, and salt water. Yeah. Okay, guys. Again, the next few slides are just really you can go through them. They're just examples of these different solutions. Okay, and so I'm going to let you just look at those for yourself. Just to speed things up a little bit. This also explains, as you go through them, why some animals, or especially for example fish, some fish are freshwater fish and some fish are saltwater fish, and you can't take one from the other because they're designed to just deal with, if they're in freshwater, then they're in a hypotonic solution. And their bodies are designed to deal with the water wanting to kind of always enter the cells, whereas a fish in salt water is dealing with the opposite situation, but their bodies are designed to deal with their cells always sort of wanting to release their water. And they just have ways to deal with that, and they wouldn't deal well if you switch them. Yeah, so again, all right. Now, I'm going to end this lesson here, and we're going to have one more YouTube lesson to finish off the chapter. And that primarily means we're going to take a look again at all the different ways that transport can happen in and out of the cell. Some of those ways don't require energy, like diffusion, and there will be processes that do. And we're going to talk a lot more about those proteins we were talking about earlier that are embedded in the cell membrane and how they play a role in transport across the membrane.